Hello everyone and welcome to another snow day edition of our Molecular Cells and Genes lecture. Today we're going to be finishing up our discussion of chromosomal inheritance and beginning our discussion of molecular inheritance, which means we're going to talk about the structure of DNA and exactly how DNA replicates. So in order to follow along with today's lecture, you're going to want to sign in to the Socrative so that you can answer a couple of questions. You'll log in just like normal, and then you'll complete the quiz as we go through. So here are the questions that I'd like you to answer at the beginning. Go ahead and answer at your own pace. Pause the video here, and then you can pick it back up again after you've answered the questions. Alright, so today we're going to be talking about linked genes. Now these are different from the sex-linked genes, which you named when you were doing our questions at the beginning of this lecture. Linked genes are two genes that are found on the same autosomal chromosome. So the linkage refers to the same idea. Linkage refers to two things that are being inherited together. Sex-linked genes are genes that are inherited in a way that links them to biological sex. But when we're talking about just straight linked genes, we're talking about two genes that are inherited together because they're found on the same piece of DNA. Now, this characteristic was also identified or fleshed out by Thomas Hunt Morgan, who did experiments with fruit flies in which he noticed a strange connection in the inheritance between two different characters. So we're going to look at two characteristics for fly body color and fly wing size. Now, gene B is going to control body color. It's named for black, which is our mutant phenotype, and that mutant phenotype is recessive to the normal body color gray, which is going to be indicated by B+. VG also stands for vestigial. Vestigial means like tiny, okay? So vestigial wings are going to be tiny wings, and this is recessive for normal-sized wings, which are indicated by VG+. So now I want you to answer this question. Which of these phenotypes represents a fly with vestigial wings and a black body? So in order to answer this question, we have to go back and read the text again. So if gray body color is normal and dominant to black body color, both of these phenotypes are going to be controlled by the B gene. The plus sign indicates for us the wild type phenotype. Wild type phenotypes are going to be the most common phenotype or the normal phenotype in a population, which our text tells us is the gray body color. So B plus is going to indicate a gray body color. Since black is recessive to that gray body color, we need to have an individual that's homozygous for the recessive B gene, so BB, with no B plus alleles. Very similar for vestigial wings, the normal phenotype is dominant. That normal phenotype is going to be normal or large wings, indicated by VG+, and the tiny vestigial wings by just VG. Since this is recessive, in order to see vestigial wing phenotype, we need to have a homozygous recessive at both of these traits. Now, this individual, which is homozygous recessive for both mutant phenotypes, is going to be perfect for us to do a test cross. Remember, a test cross is a cross where we breed an individual with a homozygous recessive, which helps us to see the phenotype or the genotype of that second parent. So let's look at an instance in which Thomas Morgan Hunt actually did this. So our look at our P generation. We use two true breeding parents, which means these are going to be homozygote individuals. We have one that's a wild type, so gray body and normal wings, B+, B+, VG+, VG+. And we have a double mutant, black body and vestigial wings, BB, VG. So these individuals bred together and created an F1 hybrid, which was heterozygous for both the B gene and the VG gene. So now I want you to draw out a test cross crossing this F1 hybrid with the homozygous recessive. It's very important that you draw out this test cross and test your ability to be able to do this. So I want you to pause the video here before you go any further and draw out this cross for this second crossing, our test cross. All right, now that you've drawn this cross, I want you to answer a question. What ratio would Morgan expect to see from this test cross? So take a look at the phenotypes of the children from your test cross and let me know the ratio of those phenotypes. So here's an opportunity for you to check your cross. Do you see something like this? Make sure that your gametes were sorted correctly. Each gamete should have one copy of each gene. They should be half of a baby, right? And since every baby gets 2 copies of each gene, that means that each gamete should have a single copy. So that means each gamete should have one of either B or B+, and one of either VG or VG+. The combinations of those is going to depend on what the parent has. Our homozygous recessive only has one possible gamete, B and VG. Both are the mutant alleles. So that means that they can only give us one of these characteristics here. This is the only possibility that could have been produced by our homozygous recessive. But our F1 hybrid generation can produce any of these combinations. So that gives us a 1 to 1 to 1 to 1 ratio, or 25% chance for all of these individuals. However, what Morgan saw was very different. What Morgan saw was that there is a significantly higher chance of inheriting the combination of these genes which matched one of the 2 parents. So you were significantly more likely to be either a double wild type characteristic fly or a double mutant phenotype fly. Having only one mutant and one phenotype of these genes was much rarer. And so this presented an interesting challenge because this sort of defied the idea of independent assortment. And the theory which Morgan came up with was that if these 2 genes were found on the same piece of DNA, then that would significantly increase their chances of being inherited together, because those 2 would no longer assort independently. So you can see here our F1 hybrid. This is going to be one chromosome, the chromosome that this individual inherited from the wild type phenotype parent, and one chromosome from its mutant phenotype parent. Since these are on the same piece of DNA, it's far more likely that these 2 will be inherited together, along with one chromosome from the other parent in the next generation, and that the 2 wild type chromosome, or the 2 wild type alleles, would be inherited together in the majority of offspring. In order to inherit those non-parental phenotypes, which were rarer but did in fact exist, there'd have to be some form of genetic recombination. Now this is great because this also explains why you don't look exactly like your mom or dad, right? Or even half of your mom and dad. You're kind of some recombination of those different traits, but we only know of one thing in this class that can produce this kind of genetic recombination. So please tell me, what is the phenomenon which we've already discussed that explains when genes from the same chromosome are not inherited together? Great! Did you tell me crossover? Because crossover is the only instance in this class in which you can get exchange of chromosomal information, producing these new recombinant chromosomes. This is the only time in which you can switch these types of DNA. Okay? Now, this is going to be the end of your Socrative quiz for today. But in order for you to get your extra credit point that counts for your attendance, make sure that you keep watching this video, because I have an extra assignment for you at the end that you'll need to turn in at the beginning of class. So here's an example of that crossover. You can see here these are the individual from the F1 generation and the 2 chromosomes inherited from the true breeding parents. These duplicate in the interphase before meiosis, and during meiosis 1 we can get crossover between the 2 non-sister chromatids that are lining up at the metaphase plate. If that happens, we get new recombinant chromosomes that explain these new non-parental phenotypes that are inherited in some of these individuals. But since crossover has to occur between these genes, it significantly reduces the likelihood of creating these new non-parental phenotypes. Now let's switch gears and talk about when something goes wrong in this process. We're going to focus on non-disjunction, particularly this concept that we call aneuploidy, but you can also read in your textbook about alterations in chromosome structure, also known as different types of mutations. So non-disjunction refers to the fact that sometimes chromosomes do not separate normally during meiosis. Now this can happen in both anaphase 1 or anaphase 2, and it changes slightly the nature of this aneuploidy in these different gametes. So non-disjunction, if you think of disjunction being the separation or the disjointing of either a pair of homologous chromosomes or sister chromatids, non-disjunction being the failure of that separation, this happens during anaphase. If this happens during meiosis 1, then what we're talking about is non-disjunction that fails to separate a pair of homologous chromosomes. Because of this, both daughter cells from meiosis 1 will be affected. One daughter cell and both of the gametes that result will be N+1 because they're haploid but with one extra chromosome. And the other, which will be missing that homologous chromosome, will be N-1. But if this happens in meiosis 2, one of those daughter cells and its resultant gametes will likely be normal, and only a single one of those daughter cells will have failure of sister chromatids to separate, resulting in a single N+1 and N-1. You'll have the opportunity to work through an example of non-disjunction in sex chromosomes using your worksheet, where you can see how these differences can result in different possibilities for the children.
See what happens when mitosis malfunctions. Newt epithelial cells were grown on cover slips and photographed using differential interference contrast microscopy. Notice how in this cell, one chromosome, black arrow, never arrives at the equatorial plate, likely due to improper attachment to the spindle fiber. As in meiotic non-disjunction, this will result in the
So this is an example of that non-disjunction, where you can see the chromosome that didn't line up and wasn't assorted correctly. Now, in the event that one of those gametes becomes fertilized, that results in a condition known as aneuploidy. Aneuploidy is when you have one chromosome with the wrong number of that chromosome. Okay, so it's no longer diploid. If you have a single copy of that chromosome, we call it monosomy. There's only a single example of a viable zygote with monosomy, that's monosomy X. And then if you have 3 copies of a particular chromosome, that's known as trisomy. There's a lot more viable numbers of, or viable types of trisomy. We're going to talk about just a couple of them. Now, if you have a complete additional set of chromosomes, that's what we call polyploidy. So polyploidy is not a single chromosome number mistaken. This is complete sets of chromosome numbers. Animals do not have viable polyploidy, but this is really common in plants. You see this all the time when you open up a container of strawberries. So this strawberry that's on the left here is an example of a normal diploid strawberry. This is a strawberry that has tetraploidy. Multiple sets of all of its genetic information leads to what looks almost like multiple organisms squished together into a single organism. Or this strawberry here, which is almost certainly octoploidy. Farmers have genetically engineered and designed and searched for plants that are able to do this because you can see they result in larger fruit. But this isn't something that's viable in humans. Down syndrome is an example of an autosomal trisomy, specifically trisomy 21. It's the only example of an autosomal trisomy that we're going to talk about, but Down syndrome is arguably a very important mental condition in the United States, and it affects about 1 in every 800 children that are born. So although we don't know exactly why, we do know that the frequency of Down syndrome increases as the age of the mother increases. So it may be that non-disjunction increases as the age of the mother increases. Now we're going to talk about non-disjunction of sex chromosomes and the possible conditions that can result. Turner syndrome, which is also known as monosomy X, is the only viable monosomy in humans. Linda Hunt is an example of a woman who has Turner syndrome. She has only a single copy of the X chromosome. Others may have part of an X chromosome, okay? So it can be all or part of a missing X chromosome that makes Turner syndrome. That tends to form this webbing on the neck here, which appears like a thicker neck. And these individuals are sterile, which means that they can't have offspring, and they do have numerous and variable health problems, often related to the severity of how much of their X chromosome is missing. They also tend to be much shorter in stature, which anyone who has seen Linda Hunt acting in NCIS Los Angeles knows she's quite a short, however feisty woman. Trisomy X, or 3 X's, occurs in about 1 in every 1,000 females, but this typically results in no disease, maybe a slight increase for a mental handicap, but most individuals don't even realize that they have this trisomy X. So you can see that even though we're talking about the same chromosome, there's big differences in the results of having one extra or one too few. Now let's talk about males. Males all have a Y chromosome. Males who have an extra X chromosome in addition, so XXY, as shown here in this karyotype, have what's known as Klinefelters. So this is an extra X chromosome, and the greatest thing is that individuals with Klinefelters are sterile. In terms of famous individuals who may have had Klinefelters, George Washington is the most famous. Nobody knows for sure, but George Washington had no children of his own, and he had a couple of other attributes that are kind of similar in karyotype specific for Klinefelters. Here's an individual who also has Klinefelters. Many of them have somewhat feminine characteristics. They may even have sort of prepubescent breasts. They also tend to be rather tall. About 1 in 1,000 males is an XYY. And these individuals, similar to trisomy X, don't typically know that they have no disease. So these individuals are not typically sterile, and they're similar to kind of that idea of there's no real linkage between any particular disease characteristic. We're going to talk about one last pattern of inheritance, which is sort of an exception to this standard Mendelian practice. So genomic imprinting is a cool example in which one parent's genes are silenced, depending on what the sex of that parent is. So that means that within the newly forming zygote, either maternal or paternal versions of that gene are specifically silenced. This has nothing to do with dominance or recessive, so the expression of a particular gene actually has to do with which parent you inherit them from. You'll talk more about these in genetics, and you can read about them in your textbook, and they're really cool examples. But I actually want to focus on these extranuclear genes, specifically the mitochondrial genes for our example here. Mitochondrial genes are interesting because when you have a fertilized egg, that egg provides all of the cytoplasm for that new zygote. Since the mitochondria carries the cytoplasm, that means that all mitochondrial genes in that baby come from the mom. Since mitochondria don't carry a whole lot of genes, it's not a major component, but as you know, since the mitochondria are responsible for producing all the ATP for that baby, those mitochondria are extremely important. Individuals who have defective mitochondria result in diseases that affect development and muscular and nervous systems as well, and can lead to disease. The picture on the right shows the very first baby who was ever born with 3 parents. This was done by using the cytoplasm of a healthy mother with healthy mitochondria, and we replace the nucleus of that cell using a nucleus of a fertilized egg from a mom who had unhealthy mitochondria and biological dad. So in this way, this is the first 3-parent baby.
The British Parliament is getting worldwide headlines this morning after an historic vote to allow the creation of babies from the DNA of 3 people. The controversial procedure is expected to cut the risk of genetic disorders. Mark Phillips is outside Parliament in London, where the new rules are moving forward. Mark, good morning to you.
Good morning. Well, it was a highly charged debate here. Phrases like designer children and playing God get thrown around, and there's some who fear this is the beginning of a slippery slope toward genetically engineering children.
Yesterday may well be remembered as the day the genetic future of our species began.
Jamie Metzl is a writer on genetics policy, and his words may sound a little overstated. But not to Poppy's family. She's almost 4 and suffers from mitochondrial disease. She can't walk and can barely speak. Her cells don't produce the energy they need for her to develop. Lily, her sister, does not suffer from the disease, but may carry the genetic defect. The procedure would help mothers whose eggs have a healthy nucleus but are surrounded by defective mitochondria, the little batteries that power cells. That nucleus is transferred to a donor egg with healthy mitochondria. An embryo produced with that hybrid egg would contain DNA from the mother and the father, plus a tiny amount from the egg donor. It's this 3-parent child that poses the ethical dilemma. But not to Poppy's family. It means the girls, Lily certainly, and Poppy, if she lives to reproductive age, could have children who don't have the disease.
It's inherited. It will pass down the female line, so to give them a choice when they come to the age of wanting, thinking about children, an option of having a healthy child.
Science won the moral argument in Britain, but in the US, the discussion gets political.
It's very, very difficult to have these kinds of conversations in the United States because we have the big divide between the so-called right to choice and the so-called right to life, that we very, very quickly get caught up in national politics that are in some ways bigger than the issue of mitochondrial transfer.
Now, it's important to say that the DNA of the so-called 3rd parent in the egg would not affect the characteristics of the child. It would, however, Nora, give that kid a chance to have a normal and healthy life.
An important distinction, Mark. Thank you so much.
So this is a fantastic example of how understanding genetics and genetics engineering has really become something that's influenced the medical field. And so in order for us to really move forward with our discussion, we need to start illustrating and defining and working with the details of how DNA actually works. So we all know that DNA is the genetic material, but how exactly was it that scientists proved this? So Gregor Mendel was the first who described this idea of genetic inheritance, and he was the one who illustrated that in fact there were genetics that we could track and use to better understand these characteristics, not only of plants, but of us. And so it was Thomas Morgan Hunt that proved this chromosomal inheritance, that there was a piece, a unit that we could track that contained these heritable units that we now call genes. In 1950, Chargaff demonstrated that the percentage of A and T nucleotides within a particular organism was always equal, and that G and C were also always equal. But he published his findings in this obscure journal. So he showed that within a cow, A and T percentages were the same, G and C percentages were the same of DNA. And that in a plant, A and T were the same, G and C were the same, and in humans, A and T, G and C. But what he couldn't explain was that between all of these organisms, the percentages were not identical to each other. And that number varied between species. And that sort of was lost until in 1952, Hershey and Chase showed that DNA was the genetic material, and that sort of gave a context to his findings. Hershey and Chase used a virus that infects bacteria. Viruses are composed of only two components, the DNA within them and the protein shell that coats them. Now, prior to Hershey and Chase, everybody thought that protein was going to be the genetic material because protein is a lot more diverse than DNA. Remember, DNA has only 4 nucleotides. That's all the variability it has. But we have 20 different amino acids that can compose different proteins. However, what they showed was that it was actually the DNA that was necessary to produce new baby viruses during the infection of a bacterial cell, and that protein coat was actually not required for this process. So DNA had to be the genetic material that held the heritable information necessary to make new baby viruses. Watson and Crick used the data from Chargaff, Hershey and Chase, as well as Rosalind Franklin to describe the structure of DNA, which is what we're going to talk about now. So DNA has a sugar phosphate backbone, and this backbone is going to be composed of both the phosphate, which you guys know comes from the triphosphate tails, as well as a sugar, which can be either deoxyribose or ribose. You can look here at this carbon, which is known as the 2' carbon, to identify whether this is ribose or deoxyribose. Since this is just a hydrogen and not a hydroxyl group, we know that this is deoxyribose. The 1' carbon always has a nitrogenous base attached, which reaches into the inside of the DNA molecule. The 3' carbon has a hydroxyl group, and this is going to be the site of the growing nucleotide chain. So this is going to be the place in which new nucleotides are added to the chain. And off the 5' carbon is going to be the phosphate group, which is linked to the next sugar. So this backbone is composed of covalent bonds, and it forms a very strong backbone. You always have to label the 5' end and the 3' end of this backbone, which is especially important because we can only add nucleotides to the 3' end because only the 3' end has this hydroxyl group. Within the molecule and holding the two strands together is hydrogen bonds. Remember, we've talked about hydrogen bonds in the past, so make sure you go back and review and you understand exactly what these hydrogen bonds are. Here is the polar bond between nitrogen and hydrogen, or carbon and oxygen, which allows for the creation of these partial charges and the hydrogen bond attraction between them. Adenine and thymine form two hydrogen bonds, which is why adenine and thymine are always found together. And it's why Chargaff noticed that there were always equal percentages of adenine and thymine. Guanine and cytosine have three hydrogen bonds. They hydrogen bond to each other between two strands, and why they are always found in equal percentages in any living thing. So DNA is a double helix made of two strands, because between that helix is hydrogen bonded either G and C or A and T. The two strands run anti-parallel because one of those strands is going to be in the 3' to 5' orientation, while the other is 3' to 5'. Note here the hydrogen bonding between the bases, the nitrogenous bases, which are reaching between and connecting these two strands. No matter how we represent these strands or how detailed the image is, we always have to represent the orientation of those strands, because that's essential for us to describing the orientation of how this DNA is being replicated. So now let's watch a video that reviews what we just talked about.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So now we're going to be talking about how DNA is replicated. And I told you that in order to get your extra credit, you needed to turn something in at the beginning of our next class. So what I'm going to ask you to do is turn in a drawing of the replication fork. So we're going to start by talking about replication as a whole, and then we're going to move into a discussion of each of the components of a replication fork. Now, unfortunately, I can't record myself drawing, but what I can do is I'm going to provide you additional videos that look at how to draw a replication fork. you'll want to draw that and turn it in to me at the beginning of class. I'll look at those drawings and let you know if I have any feedback. So make sure that you put your name on them so that you get credit. Now, when we talk about replication as a whole, we need to discuss the fact that replication in DNA is what we call a semi-conservative process. Semi-conservative tells us that of the parental molecule of DNA, that molecule is going to split, and each of those parental strands is going to be used as a template for the formation of new strands. So we're going to conserve one parental strand and add an additional new daughter strand in each of the new DNA's molecules that are created. So we call this semi-conservative replication. All replication begins at sequences known as origins of replication. Eukaryotes have many origins of replication along the length of a chromosome. Each one of these origins of replication opens up into a bubble, and from that bubble, replication proceeds in both directions. In eukaryotes, this proceeds from many origins until eventually each of those merges and forms two unique daughter DNA molecules. You can see here bubbles that have formed in a chromosome. This bubble has proceeded significantly farther than, say, these bubbles. But eventually, they're each going to merge into form two new DNA daughter molecules. Now, each side of these bubbles, each one of these arrows, is representing a single replication fork. So each fork is moving in one direction of this DNA replication. And each fork is going to be composed of a leading strand being synthesized and a lagging strand that's being synthesized. In this class, you only need to draw a single replication fork. So only ever do one replication fork at a time, but it's important that you know that this is only one half of a bubble, and that at the other side is going to be an opposite replication fork moving in the opposite direction. I will never ask you to draw a replication fork from scratch in this class, but I strongly recommend that you practice being able to draw this replication fork, because if you understand the function of all these enzymes, then you can put them into this context of being able to draw this replication fork in a way that makes sense. So, in order for us to draw a replication fork, the first thing that we need to do is draw the opening of this strand. So you'll always start by drawing double-stranded DNA on this side, and then how these open up into a single strand here on the top and the bottom. Now remember, you always have to label the strands of your DNA. So here we have a top strand that's 5' to 3', and a bottom strand that's 3' to 5'. So here's our 5' to 3', and our bottom strand, which runs anti-parallel of the top. The enzyme that's responsible for opening up this replication fork, for unwinding the DNA, is helicase. Helicase is actually going to force these two apart. The enzyme topoisomerase is responsible for untwisting the DNA, because as helicase unwinds this section of DNA, it forces this coiled DNA behind it to keep coiling tighter and tighter and tighter. So topoisomerase relieves the tension that's caused by helicase unwinding this bubble. Since DNA doesn't like to be single-stranded, we use these proteins called single-stranded DNA binding proteins to keep the fork open, to stabilize the single-stranded regions here. These three factors together are acting to set up our replication fork for us. But they're not going to be involved in the actual process of creating any of the new DNA daughter strands. The enzyme that's responsible for creating new daughter strands, for actually catalyzing the addition of new DNA nucleotides, is called DNA polymerase. DNA polymerase uses the energy that's contained in the triphosphates of these single nucleotides, and it catalyzes the addition onto the 3' hydroxyl group of an existing chain. Now this is really important. DNA polymerases can only catalyze the addition of new nucleotides onto an existing chain. But remember, we didn't have any DNA to add on to in our previous fork, right? All we did is open up 2 existing strands. We didn't have anything for these to add onto. So what we need to do is lay down a short primer. So just a couple of nucleotides. But this is going to be an RNA primer. And so we call the enzyme that lays this primer RNA primase. Make sure that you designate that these are composed of RNA. So in our figure here, they're shown as red. And we label these as RNA primers and RNA primase so that we know that these are actually composed of different nucleotides. Note that even though these are RNA, they're still going to be running in that anti-parallel direction. And that DNA can now be added by DNA polymerase onto this 3' end. So we're going to talk first about the leading strand, because since DNA can only be added onto the 3' end, there's going to be one direction towards the fork, which is really straightforward, and another direction away from the fork, which requires some additional steps because we have to synthesize DNA going in the opposite direction. We call that the lagging strand. But let's start with the leading strand. In the leading strand, we can lay down our RNA primer here, close to the origin of replication. And then our DNA polymerase can begin to catalyze the addition of nucleotides onto the 3' end of that primer, and it can run continuously. And even as our fork opens up, as helicase continues the process of unwinding DNA, this DNA polymerase can just continuously elongate in that addition, always adding nucleotides onto the 3' end. This DNA polymerase is known as DNA polymerase 3. And I always remember 3 because it's the larger number and it's going to end up doing the bulk of the work. But it's important that you always tell me the number of this DNA polymerase. And that as you draw this strand, you also need to label it as the leading strand.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The 2 strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
So now let's contrast our leading strand, which was very straightforward, with how we elongate the lagging strand. The lagging strand is going to be synthesized in a series of segments or fragments known as Okazaki fragments. Okazaki is the name of the guy who discovered these fragments, so unfortunately the name actually doesn't tell you really a whole lot about the fragments themselves. But these Okazaki fragments are all going to begin in the same way as our leading strand. So in the lagging strand, The first thing that's going to happen is that we are going to lay down an RNA primer. So RNA primase has a role here too. When we lay down that RNA primer, DNA polymerase III is going to extend that primer. In this case, DNA polymerase III can only extend until it runs into the previous Okazaki fragment, which you'll see a picture of in just a minute to explain why. But this is because we're actually extending in the opposite direction, away from the replication fork. So what happens is another RNA primer gets laid, and this RNA polymerase extends until it runs into our previous fragment. So we sort of get the creation of multiple fragments at a time. However, we have two problems that we need to solve. The first is that we now have RNA in what should be a DNA molecule, right? Our chromosomes are made exclusively from DNA. So we need to replace this RNA. So we're going to recruit another DNA polymerase, since DNA polymerases are responsible for creating polymers of DNA. This time we're going to use DNA polymerase I, and it's going to replace our RNA primer by extending this fragment and replacing the RNA with DNA. That's the role of DNA polymerase I. Now we've created new DNA, so we have all DNA in the place of this fragment. However, we still have fragments. And you can probably remember that it's not a good idea for our DNA to be in pieces. We want it to be one whole molecule. So that fragment here has a gap in its backbone. DNA ligase is an enzyme that forms the bond in this backbone. It catalyzes the reaction so that this new backbone will now have a fully covalently bonded strand. And so we go from having multiple fragments of both DNA and RNA to all DNA to a single piece of DNA.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand.
Here's an overview of what this looks like if you were to look at it all at once. And what I'd like you to do is to try and draw something similar to this. but I want you to try and draw it without simply drawing exactly what you see here. So you can go back and rewatch this video or watch another video that I'm going to post that talks about how to draw a replication fork. But I recommend that you try to do so by looking at and thinking about the functions of each of these enzymes and exactly what they're doing in this process. Now I'd like to leave you with one final thought, which is that the replication fork in a cell doesn't actually look like this, a literal fork with two train tracks running off like this. The lagging strand, which has multiple pieces in this fragment happening at one time, is actually twisted around, which allows it to better facilitate the creation of these multiple fragments at one time. It's called the DNA replication complex. And so this is an example, a more workable model of how this exactly looks and how all of these enzymes are kind of complex together and working with each other to be able to achieve this one process at any one given time. Here's an overview of each one of these enzymes for you, as well as a list of some of these terms. Make sure that you can label all of these components on a replication fork, and even better, be able to draw them all yourself. All right, thanks for listening, and don't forget to turn in that special assignment for me at the beginning of class. See you guys then.